So this is the brain, and this is again the sheep's brain, just to use an example. Now the first thing when you're doing a dissection is finding where your places are, or your landmarks. First you have to know which one's the front or the anterior, and which one is the back or the posterior. With the brain, you can know that it's the front part, and you can know where the back part is, because all you have to do is find the bulky area is. The most bulky area of the brain is the one that you can find two structures like these, which are the hemispheres, and another structure like this, which is the cerebellum. So these structures are normally found in the posterior part or the back part of the brain. Therefore, this part would be your anterior. Knowing the front and back, you can now identify the different lobes of the brain. In the front part of the brain, you have the frontal lobe, okay, followed by the parietal lobe, and then you have the occipital lobe. Okay. In humans, there is also the temporal lobe, which is on the side of the brain. In the sheep's brain, it's not as prominent as you can find it in human brains. Okay. So you have the frontal, parietal, and occipital lobe, and you also have the temporal lobe here. Okay. This will be the right hemisphere, and this one would be the left hemisphere. It also has the same number of lobes. The structure I earlier pointed to, the cerebellum, is found posterior to the cerebral hemispheres and slightly inferior. Okay. Now, we'll pause for a moment. I'd like you to identify these lobes on your specimens. The two cerebral hemispheres, the left cerebral hemisphere and the right cerebral hemisphere, are connected by the white tissue called the corpus callosum. It's usually found in the middle, above where the chambers are inside the brain. Now we're looking at the brain from the top. So this is the superior surface, or the top surface of the brain. I'll turn over the brain, so I'll show you the inferior parts of the brain. Okay. Now, in the inferior surface of the brain, the cerebellum is hidden. The superior surface is also hidden from view. What you can now see more clearly is the brainstem, okay, this white tissue here, and the undersurface of the brain. Okay, you can mainly see the temporal lobes and the frontal lobes and the occipital lobes. The parietal lobes are hidden from view. The inferior surface of the cerebellum can still be seen. The brainstem has three parts. In your specimen, you should be able to identify the midbrain, which is the topmost part of the brainstem, followed by the pons, which is the bulgy or the bulbous part of the brainstem, and the medulla, which is the most inferior part or most caudal part of the brainstem, and it's the one that's connected to the spinal cord. Now we'll leave a few moments for you to find these important structures on your sample. You should remember that the brain is encased within the skull. In this specimen, we don't have that skull or that bony structure. But around the brain, okay, you have the tissue that covers it. We call it the matter. Now, there are two types of matter, the dura mater and the pia mater. Okay. The pia mater is the tissue that is most adherent to the brain. You may find it in your specimen. Okay. Sometimes you can still find it in in certain specimens. Okay. So the tissue or the lining that is most adherent or closest to the brain is your pia mater. The thicker tissue or lining covering the brain is the dura mater. Okay. The liquid that keeps the brain floating within the skull is your cerebrospinal fluid. Okay. We don't have a sample of that one, but imagine the brain floating inside an encased inside the cranium, and the way it will float inside the skull is with the help of that CSF or the cerebrospinal fluid. This is a sample of a cerebral hemisphere. When you look at the external surface of the cerebral hemisphere, you can see these folds and gaps on the surface. The folds that you see, these folds, we call it the gyrus. Plural form is gyri, Gyri. The gaps that you find between the folds, okay, these gaps, we call it the sulcus. The plural form is sulci. Let's have a moment and find these structures on your samples.